Hello guys, I'll speak in English because I see that's the rule around here. So my name is Martina. You met Sasha online a few days ago. He gave presentation on Monday, I think. I'm Sasha colleague from Croatian National TV. We do that scientific show. And also I'm your colleague because I'm a fellow student as you are. I've just finished my master thesis. That's why I decided to talk today about something that I think might be very useful for you very soon. So you all got in one point of time those obligate seminars on university. You know what I think about when you get the paper and you have to write a seminar and give oral presentation about it. So you've done that?
Yes.
Yes, great. And there are two ways how to do that. Okay. I won't talk about writing part because there are rules, you know how to write, introduction, discussion, results, everything. I won't talk about that part. I'll talk about the other part, which is actually an oral presentation. There are two ways to do it. We've all been in this situation, okay? It's the night before, we only have 5 minutes to do a 10-minute seminar, we just put some slides on and hope for the best tomorrow. Okay, so I believe we all did that kind of presentations. But there are also another way to do it. I'll try to play this video.
Oh, Ralph, how I envy your optimism. There is no Springfield 50 years in the future. Global warming trapping the CO2 inside our poisonous atmosphere. Our superheated oceans will rise, drowning our land, leaving what's left of humanity bathing in deserts that once fed the world. Like the new night of Earth's darkness falls.
So, you've seen what Lisa done here. I'm not sure have you heard, but you've seen what she done. There are different reasons why to do a presentation like this. Maybe that particular subject is really important to you. Okay, that's something that you are really interested in, you're really keen on that subject, and you want your colleagues to understand that. Maybe you want to impress them. Maybe you want to impress your professor because you want to do your master thesis in his or her group. That's okay. You have your own reasons. Maybe you just want to practice giving oral presentations. But this is the way I usually do it. Okay, so that's why I decided to give you a concrete example of one presentation that I've done on my 4th year of studies for a class called Physical Chemistry of Micromolecules. I've heard that most of you are physicists, yes? Okay, so I believe you could keep on with my presentation. It's a 15-minute seminar, and this is how I did it. Okay, so first of all, I'll do an experiment. I have two liquids here. They are very strange, you've never seen them before. I believe you recognize them. One is pale yellow, sticky, use it for cooking. What is it?
Oil.
An oil, cooking oil, okay. And here's the other one. It has no taste, no smell. It's water. Okay, so first of all, I'll just put a drop of color here in a water glass to see the difference. Of course, my fingers are always blue after this. And so let me ask you, what will happen if I mix these two liquids together?
The blue water will go under the oil.
Great, so we already have a hypothesis, and let's now be the real scientist and do the experiment. I'll now pour in blue stuff into the yellow stuff to see what's going on here.
Yeah.
So you all expected this, yes? Okay, so here's a question from me as a chemist to you as a physicist. Why this stuff happens here?
Because the water has more density than the oil, so it falls down.
Yes, density is one reason, but there's also another reason. It's a little bit more chemistry in it.
Why don't they mix?
Why don't they mix?
Yeah,
Yes,
that's the question.
that's the real question.
Okay, come on, chemistry.
Come on, guys. What kind of interactions are there in the water?
Molecular.
Molecular, yes, but they are polar. Oil is nonpolar? It's really simple chemistry. Okay, so polar water likes polar water more than it likes oil. That's why all water molecules stick together and oil molecules stick together. And what happens here between the air and the oil? How is it called?
It's the same concentration.
There are also two different phases here, and there is a surface tension between. Yes. Okay, so it's a really simple experiment. We can make it more fun by adding this magic pill inside. And in a few moments, we will have something called, you know how it's called, this childish experiment?
Lava lamp.
It's called lava lamp, yes. So that's extremely simple experiment that I usually do for children in kindergarten. So I now need your explanation. How is it possible that experiment as simple as this one in this cup ended up in one of the best scientific journals? Here in Science, since 2009, we have some liquid droplets dissolution condensation. Here in Nature, we have phase transitions. Here in Cell, again, some condensation, dissolution, simple chemistry or physics as it happens here. So something simple as this has been studied recently in best of scientific journals. There's also one thing that you already seen. This is the eukaryotic cell. So is there anybody in the audience who might say why is the eukaryotic cell better than prokaryotic? You don't know? Okay, so I'll tell you.
Sorry.
So it's actually, the reason is that eukaryotic cell has compartments. It has membranes, so all these different little organelles that you see are divided from the cell with membrane, with lipid membrane. It's important to divide these reactions because here, for example, in lysosome, that's where proteins are being cut. When you no longer need some proteins, you cut them here in the lysosome. And pH here is around 2. That's very acidic. Cytoplasm is around 6 or 7. That's almost like water because most of the cytoplasm is water. You don't want this lysosome to burst inside the cell because it will eat whole the cell. Okay, so that's why eukaryotic cell divided these compartments. And traffic from one compartment to another is extremely expensive. There is this currency in a cell called ATP that we need to invest to move a protein from here to here. That's really expensive, but that works for the cell because we can divide these different reactions. And we were happy with this explanation, okay? Biologists for centuries thought, okay, that's all we know. There are some compartments, there is some ATP, nothing special about it. But then, they discovered a lot of different little dots around the cell that seemed that they don't have a membrane. Okay, but they have some particular chemical reactions inside of them, and they don't have a membrane that divides them from the cytoplasm. They looked like balls, like little balls, and scientists said, okay, there are some solid balls around the cell and they move around. They are solids, that's the important part. So here are example of them. Those are called P granuli. They contain a lot of RNA and proteins, which are important. So this is embryo of a little worm, and this part that has more green dots will later become a head, and this part will later become tail. So it's important for the specification of the cell. And these scientists ask themselves, how does these green dots end up in this part? So they should be little balls, and they should just roll to the other side. And they've seen this under a microscope. Please look here in this dark blue square and try to watch what happens with these little balls. It will happen somewhere around here, I think. Just try to watch, and then I'll see you. Then I ask you, have you seen it? Did you see this? And now it will happen somewhere around here.
Bigger and bigger.
they're becoming bigger. So can you explain me now, how is it possible if two balls are solid, if you push two solid balls to one another, what will happen?
They will combine.
This one will, but if you have solid balls here on the table, like volleyball and football ball, they will crash and each of them will go apart. Okay, so that happens with the solids. And this thing here happens with the liquids. So that was the only possible explanation. They pulled these little balls outside, they put them on the glass, and they look like rain on a windshield. So the only possible explanation is that in the cell we have some little parts that don't have membrane but are separated just like oil and water. Some reactions happen here, some reactions happen here, and they do not mix. It's really useful for the cell because it doesn't have to use ATP to move stuff from one part to another. And then they ask themselves what kind of proteins take part in this thing called liquid-liquid phase separation. They've found these sticky patches, which are something very particular for the proteins. And there is also another interesting thing. So biochemists hate when they cannot get a really good structure of a protein because they cannot do X-ray and there are very little information that they know about that protein. These intrinsically disordered domains can't be seen on an X-ray. And that's why they don't like them. This, for example, this green one is Rust binding domain. We know its structure, so we know its function. But for those here, wavy things, we don't know the structure, so we don't know the function. But here in this work, they found out that actually this intrinsically disordered domain is the one who enables the proteins to look like these droplets, to make the phase separation. So now they know what kind of proteins take part in that. And here's something familiar. This is actually the best thing. You recognize this stuff. So these are, let's say, simple equations from physical chemistry, soft matter physics that we know for 50 or 100 years ago. Okay. And we can use them as we use them here. So we already know the calculus that we need for a really complex system as a living cell. And that was groundbreaking. That's why we had those simple stuff in those huge magazines. Final goal is to understand this system as much as possible. For example, to get the phase diagram and to understand at which temperature, with which concentration, do we have two-phase regime, one-phase regime, and stuff like that. Because even hibernation of bears has something to do with this. Some proteins become, on a lower temperature, they become these blobs and save energy for the bears. So it's a really new and interesting concept that we know for many years now. And here now I'll show you a work from Brangewine lab from 2016 done by Feric and Vaida, postdocs at the lab, who wanted to understand one little dot called nuclei. It's where proteins are made inside the nucleus. Okay. So they found their proteins of interest very easily because they had these wavy parts and they put green fluorescent tag and red fluorescent tag on them. Now when they have a tag in a living cell, they can study behavior under the microscope. And here is a film of a living cell and what's going on inside. When the film starts, you see a lot of green little dots that become one big green inside of red. Okay, so every time they watch those proteins, this happened. Every time green goes inside, red goes outside. And here, the black thing that you actually cannot see is water in cytoplasm. Now they wanted to understand the physics behind it, so they did phase diagrams, as I showed you before, dependent on salinity, on concentration, stuff like that. But they actually wanted to understand this. Why is it always that green goes inside and red outside? So this is like first year physics experiment. Hydrophobic and hydrophilic surface. Something that's hydrophilic will make this shape, and something that's hydrophobic will try to stay away from the surface. So that's the explanation actually. The green part is more hydrophobic than the red. So that's why they go like this. Green inside, red, then water. We actually have three-phase regime. Not two-phase, but three-phase regime. And again, why is this in a Cell magazine? Because of biological significance. Things goes in this way. So first we have some RNA transcript. Then we do, in green part, some processing. I mean, not we, as a cell. And then on the ribosome, proteins are being made in the red part. So it has biological path here. There is a logic behind this. Why is it always green here, red here? And there is no membrane in between. So what is being processed just goes without actually involving some extra energy for that. It's very good for the cell. There's also a huge biological significance of the whole story, whole phase separation story, because now with these concepts from physics, we can understand more about soluble molecules, liquids, and the most important part, this transition between liquid and solid. Because it's been proven that these very severe diseases are happen because of this, because of some proteins stick together with they stick together so well that they can be soluble again, okay? So they make those aggregates, those solids that lead to these diseases. So that's why we need to understand this process very well. And I mentioned this guy earlier, it's Granuli. He's the scientist who was pioneer of this idea in 2009. And this was written in one popular magazine about the subject. So if you make a discovery and at first people tell you that it can't be right, and then they eventually switch to telling you, we knew that all along, then you're probably onto something. Because first, it seemed so simple. Some of them said it's too simple, some of them said it's too, it's unnecessary, and stuff like that. And in the last 10 years, there is no paper in cell biology that doesn't include phase separation in these huge magazines. And there's also one more take-home message here. So this guy, he does now cell biology, but he's actually a physicist. He done his master thesis about phase diagrams of steel in steel company. So he used his knowledge about phase diagrams and steel to understand biology. That's why he's such a big fish, we can say, in this field. And that's also a take-home message for us to connect, not to divide, okay, you guys are chemists, you guys are physicists, you are biologists. We need to work together to solve really complex stuff with really simple solutions. So this was the first part. This was my uni seminar. And now I need to hear your comments. So what do you think about this? Is there something that you particularly liked in this talk or didn't like?
Of course we like the exhibition.
Yes.
Okay, something special. Is there something that you will remember later in this whole story?
Well, I can tell the simple experiment with a simple experiment. You're explaining something that's happening in a cell, and the science is actually simple. You're showing you, so I can understand what is going on in the cell.
That's great. That's great. So that was that level lamp thingy. Is there something that you don't like? That's also okay. Maybe this kind of presentation is not for you, and you don't enjoy people who talk loud and do this with their hand. Oh, you don't want to say. Okay, it's okay. So I think that you all recognize parts that you liked or didn't like, and you know what is good for you and for your presentation just from these 15 minutes I was talking. But now I'll just do a little bit of tips. Actually, I'll do that background. How did I make this presentation, okay? So first of all, you need literature. Most of your professors give you some scientific paper that has all these important parts. It has summary, introduction, results, discussion, literature, everything is inside. Okay. And first instinct is to read this. But if you're studying biochemistry and physical chemistry as I did, coexisting liquid phases underline nuclear subcompartments. It doesn't mean a lot. Oops. And what do you do next? Okay, so here are the guys that actually did the work. They know every beaker, every concentration, everything that went in those experiments. They know what they did best. But this guy here knows why they did it. He was the one who actually did proposal, who got the idea. That's the corresponding author, that's the boss. Okay, so next thing you do, you Google that name. And you add this word here. So you know the difference between typical scientific paper and review paper. You know what review papers are. Okay. They're written for you. They're written for students. They always look like chapter of a school book because everything is explained in there in a simple way. Okay. So they are meant for wider audience to understand the subject. And that's why you find some good reviews about your subject. That's the first thing that you actually read. And after that, you can find in the same review articles these nice infographics, because in original paper, you'll find graphs and stuff like that are really complex to understand at first glance. If you are not particular, if that's not your particular part of studies. And then use all possible sources. So if your corresponding author or his boss maybe is someone very big in the field, there is a really big chance that you can find his or hers talks on the YouTube, okay? So that's where you go. We have that saying here that says, "Tko zna što je pisac htio reći." I mean, the best way to understand what the author meant is actually to listen to the author. And half of my infographics were actually print screens from his presentations. Okay, so you can use YouTube, Twitter accounts, Facebook accounts. These big labs, they always have these platforms to communicate with people. Use them. Also, time management. That's crucial thing in giving oral presentation of any kind. First, why I'm saying 7 minutes introduction? Why did I put so much effort on the introduction? That's where I'm going to use all the stuff that I've learned from reviews, from YouTube, from Twitter, from all these different magazines and stuff that I've read. I mentioned to you the steel thing that he actually did with his master in steel business. I've learned that in on some Twitter, how it's called?
Post.
Twitter post, yes. I've learned that in a Twitter post. That's where I found it. Okay, so you can find your funny informations that keep the audience attached here. And you do that something, first of all, you need to do something familiar. You need to make a statement or experiment or something that people in the audience already know. Because then they feel like, okay, I understand this. I can take part in this because I already seen this. This is some part of general knowledge. You should start with general knowledge, with something fun, as this little experiment, and then got to specific concepts. Also, what about the results? What about the actual paper? The actual paper here was that part when you've seen the red and green blobs. That was the actual paper. It was only 2 slides. And in the end, you have to talk about, if you want your audience to listen to you, about the application, contribution, everything that, all those take-home messages that you learned during that investigation about your particular subject. And then you can use your fun thing to round out the story. That's also important for people to get the whole concept, to get the whole idea. Okay, now about the lava lamp. You know what's Cookie Midland? I had to read Proust in high school. That was the little cookie that he ate, and every time he ate it, he remembered his childhood. Do you remember that from high school? Yes, so Cookie Midland is something memorable. That's why I had the lava lamp here. I wanted something memorable that every time you see, next time you see it, you'll remember this presentation, you'll remember Connect. Okay, you need something memorable to make an impact. It has to be simple, it has to be familiar to the people, not something extra complex that they've never seen before, but it has to be part of your personality. So think about it. I've done these experiments in kindergartens for 5 years, so I can do them whenever I want. Okay, so it has to be something of yours, some part of your personality, so you don't have to act. It has to be natural. I've done those things. I do them usually. So this is also one example. I'm actually studying. I'm a chemist. I'm studying cell division. I'm in biology field, and we want to understand how does chromosome go apart in the cell by physics. Okay, so there's a lot of things to understand this matter. And here, this was a student symposium for chemists. They invited me because I'm a chemist, but I was talking to the audience that doesn't understand my subject, so I had to make it fun. That's why I did this chromosome. I made it out of some fluffy material, and I wanted to my big statement was we need to use forces to take them apart, and that's what I did. Okay, so later that day, there were thousands of presentations, tons of presentations that day, but people remember my presentation because I was the girl with the fluffy chromosome. Okay, that's the memorable thing. It has to be something connected, something natural to you. It can be your favorite song, your favorite movie, anything you like and you can connect to your story. And now I want to show you present you a real professional and this Cookie Midland thing. That's actually my mentor. She's here in the middle, this lady in the white dress. Her name is Eva Golic, and she's a biophysicist. She was my mentor on my master thesis. And she's as elegant as she's smart. That's part of her personality. She always wears high heels. Okay, so we are studying the division, cell division, and this green thingy here is called microtubule. They're actually the ropes that pull chromosomes apart. And this is our logo. Okay? So this is our logo. Those are high heels walking on a microtubule. And everywhere in the world, when some of us, postdocs, students, master students, never mind, comes to the conference and puts this on the first slide, everyone knows that that's someone from Knowledge Lab. Okay, people remember. They have that Cookie Midland. They already know, okay, this will be about bridging microtubules. I know, I understand this. I will listen to this. Just because of these little high heels here. And there is also, I'm how much time do I have?
Just talk.
Okay, there's one more slide here. Okay. There's just one more slide here because this is pro version of memorable things, and this is extra pro version. Okay. Also, my mentor here. So this is the spindle, the same thing you see here. She made a necklace here with the spindle and all our important discoveries on it. Okay. and that's on our Twitter. So maybe you can read what it says here. Ready for ACB EMBO 2019. That's the biggest conference for cell biologists. That's where she wore this necklace for the first time in front of audience of 5,000 people and talked about the spindle, but later she posted this on her Twitter. And every time someone asks her, so what are you studying, she can pull that necklace and explain to people, we are studying this thingy here which calls protein fiber. Okay. So that's like extra poro version, something you can think of in, I don't know, 20 or 30 years after you've seen this. And that's all, guys. Thank you. I hope
Comments, questions? Usually.
It was great. I see you like Simpsons.
Yes, that's also something personal. I wanted to make the round out this presentation with Simpsons.
You touched me with Simpsons. I mean, I live with them for.
Okay, guys, I understand you're hungry. I'm really glad I was able to be with you in person because this would not work, I think, online. So this is actually all tips for live presentations. You can think of your own for online. I hope so. We will do it like this in the next year.
Okay, let's thanks again to Martina.
Thank you.